All right, ladies and gentlemen, I'm going to finish up Chapter 2, Section 4 notes for you here. Hopefully by now you have read Section 4 on your own. If you have any questions about this or the reading, just email me and I'll get back to you as soon as possible. I believe this is where we stopped off. So what we're doing here is we're looking at chemical reactions and their activation energy. All chemical reactions require some activation energy, and that is what these peaks here are on this blue line and on this red line. That's the activation energy required. What they're trying to show us here is that there is one chemical reaction that would require this much activation energy to occur without the help of an enzyme, whereas this same reaction with the help of an enzyme would only require this much energy. So clearly, it would be better and easier for the cell to go through this chemical reaction with the help of an enzyme so that not as much activation energy is required. And that's what enzymes do. Enzymes will force chemical reactions to occur faster and easier by lowering the activation energy. So if you have any questions, you might want to jot them down now. Okay. Our next slide is taking us back to the carbonic acid reaction that we had talked about earlier. So let me brief you on that real quick. Our cells tend to produce a lot of waste products. One waste product that our cells produce is carbon dioxide. This is not good for your tissues or your cells. It's toxic, all right? But it's not easy to handle. So what happens is carbon dioxide will react with water to form carbonic acid. Carbonic acid is not that much better either, but at least it's easier to process. So anyhow, carbon dioxide and water will react to form carbonic acid, and then your body can get rid of it easier. All right. This reaction can occur on its own, but it takes too long. So there is an enzyme called carbonic anhydrase, as it is written up here. You might want to highlight or circle that or something. Carbonic anhydrase speeds up this chemical reaction by a factor of 10 million, which is significantly faster, right? For the same reason that I mentioned earlier. The carbonic anhydrase will take the carbon dioxide and water and force it to react more quickly, and it's also going to lower the activation energy that's required from the cell to make this reaction occur. So it's a win-win. All right. If you have any questions about this reaction, you might want to take a moment to write them down now as well. So now we're going to take a look at the next slide. All right, here's a slightly different view of enzyme activity. This is what we call the enzyme substrate complex. It sounds a lot more difficult than what it really is. All we're trying to do is point out the enzyme here. Again, we're specifically talking about carbonic anhydrase. Okay, and if you'll notice, there's a little spot here called the active site on the enzyme. All enzymes have an active site, and the active sites are very special and very unique and very tailored to certain chemicals, and I'm going to show you what I mean here in a second. So we know that carbonic anhydrase will take water and carbon dioxide and force them to react faster. So here's the water and here's the carbon dioxide, okay? That water and that carbon dioxide are referred to as substrates or reactants, okay? Substrates or reactants. And what happens is, specifically with carbonic anhydrase, only water and carbon dioxide can fit in this enzyme's active site. Again, enzymes are very specific, and only specific molecules will fit in their active site. So carbonic anhydrase's active site is specifically designed to only fit water and carbon dioxide. So that's what we see here. So now that the carbon dioxide and the water are in the active site of the enzyme and they're all together and happy, we say that this is forming the enzyme substrate complex. All right. And then what you see next is the formation of carbonic acid. So the enzyme took the carbon dioxide and the water, it forced them to react together quickly to form the carbonic acid, and that is what we're looking at here. The carbonic acid has now been formed. All right. What happens next is also special. The enzyme, carbonic anhydrase, can now release the product, the carbonic acid, and it can let it go. And so what happens now is that the active site is now free and empty, and it can do this process over and over again. So one of the questions that you might see on your test is, are enzymes reusable? And the answer is absolutely yes, they are. Enzymes are reusable. They will catalyze chemical reactions over and over and over again. And so that's what this enzyme substrate complex diagram is trying to show us. If you have any questions, again, please make sure that you write them down so you don't forget, and that you ask me either today over email or in class. And hopefully we'll get that taken care of. I'm going to show you one more, maybe two more slides here, just to kind of drive the idea home. Here's another picture of an enzyme in the blue here. And here is well, I guess this would be more of a model of an enzyme. And here is a model of the little substrate that would fit right in here. Again, all enzymes are very tailored and very specific to their certain substrates. So that's what we're looking at here. And then we'll look at this last slide here, which is the key and lock model. All right, what we're doing is comparing enzymes and their substrates to locks and keys. As you know, only a certain key will fit in a lock. Not every key will work. It's kind of like the way enzymes work. So what we're doing is we're comparing the enzyme to the lock. The active site of the enzyme would be the keyhole, and the substrate would be likened to the key itself. Again, please make sure that you write down all of your questions and thoughts and email them to me, or you can ask me tomorrow right before we take our test. Good luck.